This is the second lecture of cell biology and tomorrow is my examination. So in the first lecture we covered the cell theory and now the topic is plasma membrane, also known as plasma lemma, and its molecular concept. So I'm going to revise it. I'm going to memorize this stuff because the memorization is a core part of biology. Okay, if you don't know the term, then how could you conceptualize that particular stuff? Okay, so topics to be covered. I'm just reading my own notes which I make in my classrooms, as you can see. Okay, so on 13th of January, the third class, plasma membrane, plasma lemma, and its molecular concept. Topics to be covered. So the plasma membrane, we talk about the eukaryotic and prokaryotic plasma membrane. What are the differences structurally? Is there any structurally differences? Is there any chemical differences? Chemical components, for example, in archaea we discussed in the very first lecture that due to the membrane composition, the archaea can have survive in the very extreme conditions. For example, if we talk about the Taq polymerase we extracted from a bacteria, they can survive in a very harsh condition. Why they survive in a very harsh condition? Because of the membrane composition they have. So then we have plasma membrane in eukaryotic cells. The plasma membrane is animals, plants, and microbes. Differences. So basically, the universal. The differences in the composition is very okay. And the most prominent question, these are the question which professors ask in the university. Okay, they are not going to ask what is plasma membrane. They're going to ask the question like these, that how plasma membrane maintains homeostasis in the cell. How it is maintaining the homeostasis in the cell. So these are the question I'm going to discuss it. Okay, so plasma membrane is a thin delicate structure. It is thin, okay, thin, and it is makes sense to have a thin delicate structure because molecular trafficking was occurring. So if the road is too much of having a traffic, then it would be a difficult for cars to pass through it. Okay, so in some book it is written that it is selectively permeable, it is a semi permeable. So what are the key things uh, which you can do? Okay, so you can write selectively semi permeable, or if you are giving objective question, if you are attempting objective question, you can write semi permeable. It is making more sense than selectively, because in the older books, in the books of the plants, you see selectively in nature, and in books of latest and animals books, you're going to see it is semi permeable. So it is kind of debate. So you can say that it is selectively semi permeable. Okay, it is selectively semi permeable in nature, and it is thin delicate structure, selectively semi permeable in nature. Why it is selectively semi permeable in nature? Because the H2O, you know H2O containing molecules, H2O solution molecules easily permeable. So selective permeable का मतलब ये हुआ कि whatever the molecule H2O soluble, so the molecules which are H2O, water soluble molecules can easily allow, the access is been allowed for them. Okay, so the H2O soluble molecules have easily permeable. They have access to cross the selectively semi permeable membrane. And why it is selective semi permeable also? Because it maintains the pressure against cell wall, for example in bacteria, turgidity, and thus homeostasis. Okay, so it's not that कि हम एक लाइन में homeostasis का जो plasma membrane का जो role है homeostasis में, the role of plasma membrane in the homeostasis, we are not going to define in a single term, because the role of plasma membrane is very dynamic. So in every step we can mention how it is regulating this, how it is, for example, how it is regular, how it is transmitting the signal, how it is having role in signal transduction, and the signal transduction also help in the overall balancing of the cell, homeostasis, the ionic balance, homeostasis. So this is how we're gonna tackle the question. Okay, with each and every example we're going to include the homeostasis feature of the plasma membrane, how it is maintaining. So selectively semi-permeable in nature, H2O soluble molecules can be easily granted the permission, so they are easily permeable, and it maintains pressure against the cell wall. For example, in bacteria, it means they maintain the turgor pressure and thus homeostasis. Function of plasma membrane, I have given the sequence: CEL, SCP, STCC, CEL, SCP, STCC, CEL, cell, SCP, CEL, cell, SCP, STCC. इसका मतलब क्या हुआ भाई? C, compartmentalization. So what are the function of plasma membrane? We're not going to read the very basic function. We're going to read some genuine function of the plasma membrane. So C, compartmentalization. EL, site of enzyme localization. EL, site of enzyme localization. So the first function is compartmentalization. The second function is EL, enzyme localization. The third function is SCP. SCP, special channel protein. It means transportation, special channel proteins. So the third function is SCP, special channel protein. Fourth one is ST, signal transduction. ST, fourth one is ST, signal transduction. So C, compartmentalization. EL, enzyme localization. Third, SCP, SCP, single SCP, protein SCP. So SCP, special channel protein, special channel protein. So you have to remember this stuff, otherwise you forget it. So SCP, special channel protein. The fourth one is ST, signal transduction, and the fifth one is CC, cell communication. Okay, and the sixth one is vehicular cytosis or transportation. You can say molecular trafficking. Okay, exocytosis and endocytosis. So the first one is C. So the first one is C, compartmentalization. The second one is enzyme localization. The third one is SCP. The third one is SCP, special channel protein. The fourth one is signal transduction. The fifth one is cell communication, and the sixth one is vehicular transportation. Vascular transportation, and exocytosis and endocytosis. You can say vehicular, vesicular transportation, cytosis, vesicular cytosis. Many type of vesicles was there. Protective layer in which they have enzymes, proteins, blah blah stuff. So what are the function of plasma membrane? The first function is compartmentalization. So the membrane unbroken, continuous, enclosed all content. So what does it mean by the compartmentalization? It is membrane unbroken, continuous structure. Okay, membrane unbroken, but there are few examples, the exceptions, where is the membrane is little bit of broken. For example, in nucleus we have nuclear porins, nuclear channels we have, so they have in the discontinuous, not membrane unbroken, continuous. Okay, but majority are, and encloses all content. Okay, so that is the benefit of compartmentalization. Exception, discontinuous in some organelle. For example, nuclear membrane. Okay, we have nuclear pore, so they have discontinuous. And membrane of some organelles, for example mitochondria, they have porins. इससे क्या होता है, कंपार्टमेंटलाइजेशन से क्या होता है? First of all, you have to remember membrane unbroken continuous structure, but there are few examples. For example, nucleus. What is doing compartmentalization? Efficiency बढ़ा रहा है, division of labor को promote कर रहा है. Division of labor within cell is promoting, and it increases the efficiency. That is the first, compartmentalization. The second one is the plasma membrane is the site of enzyme localization. Okay, so you have to make this diagram. Okay, while you explain, you have to make this diagram. This is compartmentalization. It is separating from the cytosolic area from the vehicular matrix, vesicular matrix. Okay, so it is separating, so you can make it. Vacuoles also do have hydrolytic enzymes. Okay, so you can make the diagrams like this. Okay, the second one is enzyme localization, site of enzyme localization. So the plasma membrane is site of enzyme localization also. It means enzyme localization, it means that the Biochemical activities, biochemical activities, enzyme production, energy, enzyme production, energy, ATP was there, and regulate by the plasma membrane, indirectly help in the homeostasis. So the site of enzyme localization, it means the biochemical activities, the enzyme ATP production, ATP in prokaryotes, the plasma membrane has a lot more features. The mesosomes is the extension of plasma membrane. The plasma membrane have embedded ribosome. The plasma membrane have some pigments which help in photosynthesis. It is all on the plasma membrane in prokaryotes. Okay, so they have a greater role. So enzyme localization, biochemical activities regulated by the biochemical activities regulated by the plasma membrane, and these biochemical activities, the enzyme localization help in homeostasis. Okay, and it is also in help in the production of ATP. The third one is transportation. The third one is special channel protein. Special channel protein help in transportation, help in solute movement, not solute, solute from low H plus ion concentration to high H plus ion concentration. So special channel protein help in the solute movement, because only the water soluble molecules can cross this gate. So the special channel protein help in the solute movement from the low H plus ion concentration to the higher concentration. Okay. The third one is signal transduction. So the plasma membrane act as a receptor. So the plasma membrane act as a receptor to transmit information extracellular environment से intracellular environment तक, ठीक है, और neighboring cell तक. So the plasma membrane act as a receptor to transmit information extracellular, intracellular, or towards the neighboring cell, help in signal transduction. Receptor ligand molecules. Okay, so the receptor ligand molecules present in the plasma membrane, and they help in the transmission of the information. The fifth one is cell communication. You know it definitely. And the sixth one is vesicular cytosis transportation. Exocytosis, endocytosis. We have pinno and phagocytosis. So I'm revising the diagram. You should make plasma membrane double. Okay. So starting from the lower, we make like that, heterodyne vacuoles, then we have compartmentalization, then we circle like that, we have enzyme localization, biochemical activities, helping homeostasis, we have IP3 molecules, IP3 molecules is helping signal transduction, then we have receptor, then we have Na plus, H plus, and that helping special channel protein, because the special channel protein uses H plus ion. Okay, so H plus वहाँ पे utilize हो गया, and then we have cell communication. So this is the function of plasma membrane. I'm showing you. Okay, so this diagram, and cell communication, we have IP3 molecules, we have signal transduction, Na plus, H plus, and H plus is important for the special channel protein, and then we have enzyme localization, and yeah, obviously covered the compartmentalization, and this is I have written the vesicular cytosis. Okay, exocytosis, endocytosis, pinocytosis, phagocytosis, blah blah blah. So there are a lot more features also, but these are the main feature. Okay, and then we shifted to the composition of plasma membrane. That is very obvious things to talk about composition, because the composition is a very important factor of the plasma membrane and any biological living membrane. So the chemically, the plasma membrane is made up of three things. The first one is lipid, the second one is protein. So example, if I am giving in human RBC, why the scientists taken, first of all, why scientists taken the human RBC? They can take another organelle also. Why? Another cell also. Why the scientists take human blood? वो भी mature blood. There are always a reason inside. There's always a concept, because the mature RBC lack the organelle. They also have reason why they're lacking the organelle. Majority of organelle was lacking. Why? Because they want to transport a lot amount. They want to have a lot of space for the hemoglobin. That is why they exclude all the organelles. Okay, so there is no other organelles in the RBC, only the RBC and the upper layer, the plasma membrane. Then if we scientifically study the plasma membrane, then there is no hindrance, because the organelle itself also have, we see compartmentalization, they also have the layer, bound membrane layer. So and they also have the membranous layer, they also going to have membranous composition. And if we take analysis, then that composition going to hinder that upper plasma membrane layer. So that is why the scientists take the RBC. Okay, so in general, if we talk about the human mature RBC, 40% is lipid and 60% is protein. It means the solute transportation is very important in RBC, and that is why the RBC has 60% of protein and 40% of lipid, but that may vary species to species. So the composition, the chemical composition, lipid and protein varies species to species. Okay, and the third one is carbohydrates. In carbohydrates, the carbohydrates never be alone, it is always be with something, whether protein or lipid, not alone. For example, glycoprotein, glycolipid, not alone. And that carbohydrate, glycolipid, glycoprotein never be the single isolated, help in cell recognition and communication. So the cell recognition, when we go in the immune topic of immunization, topic of immune system, when we go into the detail of topic of immune system, then we going to see the role of carbohydrates, the glycoprotein and glycolipids. Okay, I in the next semester, maybe in the next, and then next semester we going to have syllabus of subject of immune system. Okay, so the carbohydrates are glycoprotein and glycolipid, and they help in cell recognition, we obviously see. So we see the fifth point, that cell communication. So how the communication is happening? So the plasma membrane do have this, the carbohydrates, so they are helping in cell communication. And the lipid protein also vary species to species. Membrane lipids are amphipathic. Okay, so the membrane lipids are amphipathic. Okay, amphipathic, why it is amphi का मतलब होता है टू, जैसे हम बोलते हैं एम्फीबियन, एम्फी मतलब एम्फीबियन मतलब कि टू, एम्फी मतलब टू, दोनों जगह रह सकता है, पानी में भी रह सकता है, लैंड में भी रह सकता है. ओके, सो द membrane lipids are amphipathic. Okay, so the lipids of the plasma membrane, जो membrane की जो lipid होती है, that is amphipathic. Okay, it means they have dual nature. The first is they do have a hydrophilic portion and hydrophobic portion, and that is why, you know, amphipathic means they have hydrophilic and phobic. Phobic means non-polarity and philic means polarity. So that is what it means by the amphipathic molecules. The membrane lipid are amphipathic. Why it is amphipathic? Because the polar head is hydrophilic, that is why they are polar, and the tail is hydrophobic, which is non-polar. So they have both tendency. They do have tendency of polarity in the head, and they do have tendency of non-polarity, hydrophobic property in the tail. Okay, so that is why they are called amphipathic molecules. So the plasma membrane lipids are amphipathic, proteins are not, lipids are amphipathic. Okay, now we see the components and composition of plasma membrane in detail. For example, we are going to see first lipids, composition, components of lipids. Okay, so we have three types of lipids. Okay, the first one is phosphoglyceride. Okay, we have phosphate attached to it, and whenever we have phosphate, we must think of, you know, signal transduction and those kind of things, you know, phosphate if we have. So the first one is phosphoglyceride, the second one is sphingosine-lipid, phosphoglyceride, the second one is sphingosine-lipid, and the third one is sterol, and for example cholesterol only in animals, you know, and in lower organism we have prokaryotes, prokaryotes. सो मेम्ब्रेन कॉम्पोजिशन में सबसे पहले लिपिड्स की बात करेंगे. फॉस्फोग्लिसराइड. इन फॉस्फोग्लिसराइड वी हैव टू कंपोनेंट्स. द फर्स्ट वन इज पीसी एंड पीआई. पीसी मीन्स phosphatidyl choline and PI means phosphoinositol. So in phosphoglyceride we have PC, phosphatidyl choline. So the name is the name is always very हार्ड इन बायोलॉजी टू रोट. ओके, सो नाम बहुत हार्ड होते हैं बायोलॉजी में, मतलब हार्ड नहीं होता है बट दे आर अजीब, कॉम्प्लेक्स टाइप ऑफ, नॉट इवन कॉम्प्लेक्स बट रेयर दैट अननोन, वी नेवर heard it like that, so it is difficult for us to remember, but you can remember as much as you read. Okay, so in phosphoglyceride, the three components of lipid: phosphoglyceride, sphingolipid, and sterols. In phosphoglyceride we have PC, phosphatidyl choline, PHOS, PHATIDYL, CHOLINE, phosphatidyl choline. The second one is phosphoinositol, phospho, PHOS, PHOS, inositol, inositol, phosphatidyl choline, PC, phosphatidyl choline. They are the most abundant phospholipid in eukaryotic membrane. So the most abundant phospholipid in the eukaryotic membrane is phosphatidyl choline, and major structural lipid of plasma membrane is phosphatidyl choline. So the major lipid of plasma membrane is phosphatidyl choline, and most abundant phospholipid in animal cell is phosphatidyl choline. Then we have inositol, minor but major signaling molecules. So they are minor in number in compared to phosphatidyl choline, but they are having extreme role in signal transduction, signaling molecules, and involved in signal transduction. So PI, phosphoinositol, IP3, we saw it in the function of plasma membrane. I've made the diagram. Okay, so they are minor role in, but structurally they have minor role, but they have great role in molecular signaling and involved in signal transduction. PI derivatives हमने previously देखा, डायग्राम में था. PIP2 से PIP2 is going to be cleaved, और PIP2 से बनेगा IP3 and DAG, which is secondary messengers. So secondary messengers are very important in the signal transduction. So PI derivatives plays central role in cell signaling pathway. So in phosphoglyceride we have phosphatidyl choline, major structural lipid in of the plasma membrane, and then we have phosphoinositol, which has major role in signaling. We have PIP2, then cleave to IP3 and DAG. Okay, then we have sphingosine lipids. The second one is sphingosine lipid. In this, phospholipid is associated with sphingomyelin. Sphingosine lipid, the lipid is associated with sphingomyelin. You know, जहाँ पे भी brain neuron आ जाए, sphingosine lipid मार के आ जाओ, ठीक है? So the sphingosine lipid in this phospholipid associated with sphingomyelin. Okay, myelin neuron याद आ रहा होगा. Okay, and we have cerebroside and ganglioside. So you just read, draw के sphingosine lipid, and this phospholipid is associated with sphingomyelin, and when we have cerebroside and ganglioside, cerebroside में लिख दो cerebroside and ganglioside. So अगर क्वेश्चन आ जाए कि cerebroside ganglioside में कौन सा लिपिड होता है, तो sphingoside लिपिड होता है जो कि sphingomyelin से बना होता है, ठीक है? नर्व ब्रेन से रिलीज, यही अगर नर्व ब्रेन से रिलेटेड कोई भी क्वेश्चन आएगा, ठीक है, तो सेरिब्रोसाइड मार के लियो, ठीक है, स्फिंगोसाइन लिपिड, ठीक है, essential for nerve cell functioning. So a sphingosine lipid is essential for nerve cell functioning and nervous system plasma membrane bridging. तो क्वेश्चन ऐसे आ सकता है 0.25 मार्क्स का कि नर्वस सिस्टम का जो प्लाज्मा मेम्ब्रेन है, नर्वस सिस्टम जो है का जो मेम्ब्रेन है वो किस चीज़ में रिच है, तो स्फिंगोसाइन लिपिड में रिच है। अगर स्फिंगोसाइन लिपिड नहीं लिखा है, स्फिंगोमाइलिन लिखा है लिख दो, वो नहीं लिखा है, अगर ऑप्शन में सेरिब्रोसाइड है, गैंग्लियोसाइड है लिख दो, ठीक है, बट ये आएगा कि कौन से लिपिड में वो अबंडेंट है, तो स्फिंगोसाइन लिपिड में अबंडेंट है। द थर्ड कॉम्पोजिशन ऑफ लिपिड ऑफ द प्लाज्मा मेम्ब्रेन इज स्टेरॉल, ठीक है, स्टेरॉल वी टॉक अबाउट द कोलेस्ट्रॉल जो कि माइनर लिपिड है, ठीक है, प्लाज्मा मेम्ब्रेन में। स्टेरॉइड लिपिड है, ठीक है, दिस आर स्टेरॉइड लिपिड फिल्ड इन स्मॉल गैप ऑफ फॉस्फोलिपिड स्ट्रक्चर ऑफ प्लाज्मा मेम्ब्रेन। तो प्लाज्मा मेम्ब्रेन का जो फॉस्फोलिपिड है जो कि मेजर पार्ट को एलोकेट करके बैठा हुआ है, तो उसमें जो छोटे छोटे गैप होते हैं वहाँ पे आता है कोलेस्ट्रॉल, और कोलेस्ट्रॉल रेगुलेशन का काम करता है। जैसे कि सीज़न वेदर चेंज होता है, जैसे ठंड आती है, गर्मी आती है, तो वहाँ पे ये मदद करता है, ठीक है, ये मेम्ब्रेन एंटी फ्रीजिंग एजेंट की तरह काम करता है, ठीक है, मेम्ब्रेन एंटी फ्रीजिंग एजेंट। तो ऐसे क्वेश्चन आ सकता है कि विच कंपोनेंट ऑफ प्लाज्मा मेम्ब्रेन हेल्प इन वेदर चेंजेस, या फिर मेम्ब्रेन एंटी फ्रीजिंग एजेंट की तरह कौन काम करता है, दैट इज स्टेरॉल, योर स्टेरॉल, द कोलेस्ट्रॉल, ओके। एंड प्रोकैरियोट्स में आप होपिनॉन्स मानते हैं, ठीक है, बहुत कम होते हैं ये, बिकॉज रेगुलेशन का काम है, तो रेगुलेशन वाली चीज़ें बहुत कम होती है, जैसे एंजाइम हो गए, ये बहुत कम होती है चीज़ें, ठीक है। रेगुलेट मेम्ब्रेन फ्लूइडिटी, अगर टेम्परेचर ज़्यादा होगा, ठीक है, तो ऑब्वियस सी बात है, अगर टेम्परेचर ज़्यादा हो गया तो फ्लूइडिटी कम हो गया, और अगर टेम्परेचर कम है, ठीक है, अगर टेम्परेचर ज़्यादा रहा तो ये फ्लूइडिटी को कम कर देगा, बिकॉज अगर टेम्परेचर को बढ़ाओगे ना तो मॉलिक्यूल्स के काइनेटिक एनर्जी बढ़ जाएंगे प्लाज्मा मेम्ब्रेन के, तो फ्लूइडिटी ज़्यादा हो जाएगी प्लाज्मा मेम्ब्रेन की। तो अब ऐसा थोड़ी है किसी को मतलब कि आजू बाजू आने जाने देगा। तो कोलेस्ट्रॉल क्या करता है, जब टेम्परेचर हाई जाता है, ठीक है, गर्मियों का मौसम आ गया, टेम्परेचर ऊपर गया, तो फ्लूइडिटी को कम कर देता है, ठीक है। और क्या करता है, अगर टेम्परेचर लो चला गया, ठंड का मौसम है, ठीक है, तो फ्लूइडिटी को ये क्या करेगा, इंक्रीज करेगा, बिकॉज ठंड में मॉलिकुलर काइनेटिक्स जो है वो कम हो जाती है, तो फ्लूइडिटी कम हो जाएगी प्लाज्मा मेम्ब्रेन की। तो ठंड के मौसम में जब टेम्परेचर कम होता है, फ्लूइडिटी को बढ़ा देता है ये कोलेस्ट्रॉल, ठीक है। तो अब तो तुम यहाँ से बोल सकते हो कि दैट इज़ हियर, ये होमोस्टेसिस में मदद कर रहा है। ये एक बहुत अच्छा पॉइंट है होमोस्टेसिस के लिए, कि जो इन्वायरमेंटल चेंजेस आ रहा है हमारे इन्वायरमेंट में, लेकिन सेल के अंदर का इन्वायरमेंट जो है वो इंटैक्ट बना हुआ है। क्यों इंटैक्ट बना हुआ है, एंटी फ्रीजिंग मॉलिक्यूल दैट इज कोलेस्ट्रॉल विच इज़ अ पार्ट ऑफ लिपिड ऑफ अ प्लाज्मा मेम्ब्रेन, इसकी वजह से टेम्परेचर हाई जा रहा है, फ्लूइडिटी कम कर दे रहा है, टेम्परेचर लो जा रहा है, फ्लूइडिटी बढ़ा दे रहा है, बिकॉज जब लो जाएगा टेम्परेचर तो फ्लूइडिटी कम हो रही है, टेम्परेचर हाई जा रहा है तो फ्लूइडिटी भी हाई जा रही है, तो वहाँ पे कम कर रहा है और यहाँ पे बढ़ा रहा है, ठीक है। फॉस्फोलिपिड मॉलिक्यूल्स ऑफ कोलेस्ट्रॉल आर लिंक्ड विद ईच अदर विच नॉन कोवेलेंट बॉन्ड, तो दिस इज वेरी इंपॉर्टेंट। मेजॉरिटी जो तुम मार के आओगे नॉन कोवेलेंट बॉन्ड ही मार के आओगे, लेकिन कहीं कहीं कोवेलेंट है, जो कि आगे देखूंगा मैं, ठीक है। So the phospholipid molecule of cholesterol, so the phospholipid molecule of cholesterol are linked with each other with non-covalent bond, ठीक है। So the phospholipid molecule जो है कोलेस्ट्रॉल का, so the कोलेस्ट्रॉल के जो molecules अगर वो लिपिड से अटैच है या फिर एक दूसरे से अटैच है, तो ये नॉन कोवेलेंट बॉन्ड के जरिए अटैच्ड है, ठीक है। तो ये एक पॉइंट मार दो, एक एंटी फ्रीजिंग का पॉइंट मार दो, और ये छोटे अमाउंट में होते हैं गैप में, जैसे पॉइंट पॉइंट करके लिखो, टीचर को भी आसानी होगी चेक करने में, ठीक है। देयर फंक्शन रैंडम लेटरल मूवमेंट कैन नॉट मूव फ्रीली, ठीक है, तो लेटरल मूवमेंट ही करने में मदद करता है, ये फ्रीली भाग नहीं सकता है, ठीक है। प्रेजेंट ओनली इन यूकैरियोट्स, प्रोकैरियोट्स में नहीं होता है, वहाँ पे होपेनॉइड्स का कॉन्सेप्ट आ जाता है। क्या माइटोकॉन्ड्रिया में होता है, एंडोसिम्बायोट थियोरी के अकॉर्डिंग देखो, माइटोकॉन्ड्रिया तो प्रोकैरियोटिक चीज़ें थी। तो दिस इज क्वेश्चन फॉर यू, ओके, एब्सेंट इन प्लांट्स एंड बैक्टीरिया, ओके। तो यहाँ पे मैंने एक डायग्राम छाप रखा है, ठीक है, यू कैन सी। सो दिस इज़ द कॉम्पोजिशन, लिपिड कॉम्पोजिशन, ठीक है। तो एक बार मैं रिवाइज कर लेता हूँ इंपॉर्टेंट टॉपिक्स को, आँख को बंद करके। लिपिड कॉम्पोजिशन में पहले तो एम्फीपेटिक हमने देख लिया था, पोलर नॉन पोलर दोनों है। और लिपिड कॉम्पोजिशन में फॉस्फोग्लिसराइड वी हैव फॉस्फेटिडाइल पोली which is most abundant structural, and in the we have phosphoinositol which is not most abundant in structural point of view but help in the molecular signaling, signal transduction. PIP2, IP3, DAG. Okay, then we have sphingolipid which is made up of phospholipid made up of sphingomyelin. We have cerebrocyte, ganglioside. Okay, जहाँ पे नर्व भाव ये लगा दो, ठीक है, और कुछ तो था नहीं नर्वस सिस्टम में जो भी है। देन वी हैव स्टेरॉल कोलेस्ट्रॉल विच इज़ स्मॉल गैप में होता है, ठीक है, लेटरल मूवमेंट कराता है, लेटरल मूवमेंट ही करता है, नॉन कोवेलेंटली अटैच्ड होता है, एंटी फ्रीजिंग मॉलिक्यूल्स है। टेम्परेचर अगर ज़्यादा जाएगा फ्लूइडिटी कम कर देगा, टेम्परेचर कम जाएगा फ्लूइडिटी बढ़ा देगा। ओके, एंड या दैट इज़ द मेन पॉइंट्स। उसके बाद हम देखेंगे मेम्ब्रेन प्रोटीन। तो हम 3 प्रकार के प्रोटीन देखेंगे। मेम्ब्रेन प्रोटीन की बात करें तो